have a question, just raise your hand. Okay, everyone, this is the introduction for Lab 9, extraction and purification of plasmid protein. So this is the green fluorescent protein that we made when we transformed the bacteria. Okay, so now we're going to go ahead and extract it and run it through a gel. But instead of being PCR where it's DNA, we're doing protein, okay? All right, green fluorescent protein originally comes from the jellyfish Aquaria victoria. It's a relatively moderate-sized protein with 238 amino acids and fluoresces green when exposed to UV light. In fluorescence, a photon of light is absorbed by the fluorophore, and as the fluorophore relaxes to the ground state, light is emitted at a different wavelength, hence the fluorescence you see. GFP absorbs light at 395 nanometers and emits green light at 475 nanometers. Okay, so the way that electromagnetic radiation works is just in wavelengths. So this is the visible spectrum. So if you see 400 nanometers, you're going to see blue, violet light. If you're seeing 500 nanometers, you're going to see green. Anywhere around 600, you're going to see orange. And anywhere around 700, you're going to see red. Okay, so the way that it works is a fluorophore is just like a protein. In our lab, we're using GFP protein, which is a fluorophore, and it absorbs light at 395 nanometers, and it emits light, or it gives off light at 475 nanometers. So it absorbs it close to 400, which means that it absorbs blue light, and then it gives off almost 500, so it gives off green. And that's why when we look at GFP under the microscope, we see green. Okay, GFP has a multitude of uses in molecular biology. It is often used as a reporter protein to indicate the levels of gene expression from a particular promoter in the nucleus. So for example, if we want to know about this specific protein called, let's see, we can call it Hedwig. Okay, because I'm a Harry Potter fan. So I found this protein and I'm going to name it Hedwig. And I want to know how much Hedwig is expressed in my sample. So what I'm going to do is I'm going to add the GFP sequence at the beginning of it, and then I'll go ahead and go along with my experiments. So when I'm looking at the results in the microscope, if I see a lot of green, that means that a lot of Hedwig was expressed. But if I don't see a lot of green, then a lot of Hedwig was not expressed. So this is how scientists use GFP as a reporter protein. It is also used as a fusion protein marker, which allows for the localization of a second protein, the fusion partner in the cell. So this information right here, you can use it for your background in your introduction. So I usually recommend 2 paragraphs in your introduction. And so you can talk about the electromagnetic spectrum, visible light, and you can get a couple of references. Any website that you use, I prefer that you do not use Wikipedia, because there's people who go on there and write the wrong information and they think it's funny. So definitely don't use Wikipedia, but any other website, .org or .edu that you use, even if you're going to get one definition from it, you can use that as a reference, okay? Actually, you're supposed to. You have to use it as a reference. So yeah, that's all your first paragraph for your intro, just a few sentences. All right, so in this experiment, you will purify GFP from E. coli cells transformed with P-glow by hydrophobic interaction chromatography. Okay, everyone, so it's going to be really important to understand a few concepts so that you know how your column works. So this right here is our electronegativity table. So it has all of the elements, and then it gives you an electronegativity value. So for example, for oxygen, the electronegativity value is 3.5, and for hydrogen, the electronegativity value is 2.1. So Okay, so now we're talking about whether something is hydrophobic or hydrophilic. So hydrophobic means that they don't like water. Well, water is polar. Water has oxygen and two hydrogens. The hydrogens are positively charged and the oxygen is slightly negatively charged. So all that means is that oxygen doesn't like to share electrons. It likes to keep most of the negative charge from the electrons. And the hydrogens, they don't really care for the electrons all that much, so that's why they're more positively charged, and oxygen is more negatively charged. So the way that we determine whether any molecule is polar or nonpolar is we look at the electronegativity values on the table that I just showed you. So for oxygen, it's about 3.4. For hydrogen, it's about 2.2. So all you do is subtract the two. So 3.4 minus 2.2 equals 1.24. So then we look at this electronegativity difference, and if the difference is more than 0.4, then your molecule is polar, or that bond is polar. So this is more than 0.4. It's 1.24. So this is really polar. Then we look at our amino acids that make up proteins. So we have 20 amino acids that make up proteins. And these amino acids have different side chains. So some of these side chains are polar and some of them are non-polar. And then some of these side chains also can have charges. So if we look here, we have positively charged amino acids, arginine, histidine, and lysine. So they have an H3 with a positive here, and then over here you also have a positive. And then we have negatively charged amino acids like aspartic acid and glutamic acid. So those are the negative charges there. So according to our lab, the GFP protein is mainly hydrophobic, so it is afraid of water. And that just means that a lot of the amino acids that make up so that just means that a lot of the side chains that make up, for example, let's say that GFP is made up of 1,000 amino acids. So we can say that maybe like 600 or 700 of them are nonpolar. They have nonpolar side chains. And so GFP is a nonpolar amino acid or protein. So then what we're doing in this lab is we're using this column, and this column has little beads. Well, these beads are nonpolar. So whenever we break up our cells and then we go ahead and put in the liquid or our supernatant into this column, what's going to happen is that our GFP protein is going to bind to these nonpolar beads because our GFP protein is nonpolar and the beads are nonpolar. So everything that is polar or hydrophilic is going to go down, and we're going to go ahead and collect it in tubes 1 and 2. And whenever we get to just our GFP protein, when that's the only thing left, that's when we're going to go ahead and collect our GFP protein in tube number 3. Okay, so now I explained how everything works, so I'm gonna go ahead and read the rest of the introduction to you. In this experiment, you will purify GFP from E. coli cells transformed with P-glow by hydrophobic interaction chromatography. GFP contains many hydrophobic amino acid residues and is therefore more hydrophobic than other proteins in the cell. In the first step, you will pass the supernatant containing the bacterial proteins and GFP over an HIC column in a highly salty buffer. So HIC stands for hydrophobic interaction chromatography column. And the reason that we use a high salt concentration is because the salt causes the three-dimensional structure of the proteins to actually change so that the hydrophobic regions of the protein move to the exterior. So we want that. We want the hydrophobic regions of this protein to be on the outside so that they can more easily bind to the hydrophobic beads that we have in the column. Okay, so let's see. The chromatography column contains a matrix of microscopic hydrophobic beads. When your sample is loaded, when the sample is loaded onto the matrix in very salty buffer, the hydrophobic proteins should stick to the beads. The more hydrophobic the proteins, the tighter they will stick. The more hydrophilic the proteins, the less they will stick. As the salt concentration is decreased, the three-dimensional structures of proteins change again so that the hydrophobic regions of the protein move back into the interior and the hydrophilic regions move to the exterior. So the first buffer that we use is going to have a lot of salt, and then the second buffer will have less salt, and then the third buffer will have like even less salt, or something like that. The first one will have a lot like that. The last one will have a lot less salt. Okay, so the last thing that I wanted to explain was about ions. So for example, any ion that you see is hydrophilic. So water loves it because you have, let's draw here. So you have oxygen, which has like a negative charge, and then you have two hydrogens, and this is one water molecule. So whenever you have an ion, which is anything that has a positive charge or a negative charge, like this potassium, sodium, or this chloride, then you can go ahead and kind of like not really attack it, but bind to it. So let's say that potassium is right here and it has this positive charge. So the oxygen is going to be attracted to it. So it's going to go ahead and bind to it. And then like another oxygen molecule on a different water molecule will also be attracted to it. So that's how water dissolves salts, because it's just like ion interactions. Okay? So that's why this stuff is hydrophilic, anything with ions like that. Okay, so for next week, after isolating our GFP protein by column chromatography, you will further purify the protein through polyacrylamide gel electrophoresis. Okay, so now I'm reading section C. Last week you purified GFP by column chromatography. Now we will further purify GFP by SDS Page. In this technique, proteins in your column fractions are first denatured by a detergent, sodium dodecyl sulfate, or SDS. SDS, which is negatively charged, coats the protein throughout, giving each protein a uniform charge to mass ratio. Proteins treated in this way can be separated on the basis of size by polyacrylamide gel electrophoresis, or PAGE. Next, we will visualize the proteins by staining the gel with Kumasi Brilliant Blue, a dye that binds tightly to proteins. GFP is approximately 27 kilodaltons in size. We should be able to identify GFP based on its position in the gel relative to size marker proteins of known size. Furthermore, the results of this experiment should allow us to assess the degree of purification of GFP relative to the crude lysate. So we're going to have a ladder, and basically we're looking for any line that goes next to 27, because that's how heavy GFP is.